Our second speaker for this afternoon is Andrew Goryachev from Edinburgh. So, welcome. Excitable systems.
Thank you very much. I would like to start by thanking the organizers for putting this very interesting interdisciplinary meeting together. I am a computational cell biologist, and we are interested in cellular morphogenesis and pattern formation. In particular, my group is interested in understanding the role of small rho GTPases as intracellular morphogens. I have to warn you that my group is completely in silico, which means that the credit for all the beautiful experimental data that I'm about to show you goes to my collaborators, Bill Bement at the University of Wisconsin-Madison, and George von Dassow at the University of Oregon. So the fundamental problem in cellular morphogenesis is how to position the plane of cell division in such a way that the genetic material is divided between two daughter cells in a proper fashion. It has been for a long time known that in animal cells, positioning of the cytokinetic furrow is determined by the location of the mitotic spindle. Already back in 1960s, Ray Rappaport in his experiments demonstrated by shifting the spindle physically using needles that if you move the spindle inside the cell, it will induce a new cytokinetic furrow at a new location, and the old furrow will be abandoned. So to explain this result, there has been two major hypotheses proposed. The so-called equatorial activation model postulates that there is a positive signal that is emitted from the central spindle, so which is this bundle of microtubules that overlap, that induces the furrow at the equator. The alternative hypothesis is the so-called polar relaxation, which postulates that the ends of astral microtubules emit some kind of negative signal that suppresses furrow formation. The truth being said is that the two hypotheses are not necessarily mutually exclusive. About a decade ago, there has been a major breakthrough in our understanding of how spindle controls the cytokinetic furrow. So what you see here is the movie by Bill Bement and George von Dassow, and what they found by using a novel at the time reagent, which is the fluorescent reporter for the activity of the small GTPase rho A, is that prior to ingression and of the cytokinetic furrow, a very dynamic zone of activity of rho GTPase forms on the equatorial cortex of the cell. So how does rho A gets activated on the equatorial cortex? Thanks to a wonderful talk from Anne this morning, I don't have to repeat the entire biology of small rho GTPases. Suffices it to say that rho A, like many other GTPases from the family of rho GTPases, in its inactive GDP-bound form, can shuttle between the cytoplasm and the membrane. And once it's on the membrane, it is activated by the class of molecules which are called GEFs into an active GTP-bound form. In that GTP-bound form, it will interact with the actin polymerization machinery and will help to contract myosins, right? So basically, the question is now transformed. How does the GEF for GTPase rho ends up on the equatorial cortex of the cells? I must say that the major GEF that contributes to the activity of rho cytokinesis is called EC2. It's also known as pebble in Drosophila. So how does the EC2 form this belt-like structure on the equatorial cortex? This has been majorly answered by the work from groups of Michael Glotzer and later Mark Petronsky, who demonstrated that ACT2 is released from the nucleus in the prophase of the cell cycle and remains pretty much cytoplasmic until the end of the cell cycle, the anaphase stage, so-called anaphase stage, when it hitchhikes on a molecular complex which is called central spindlin, and together with central spindlin it moves towards the plus of ends of microtubules, accumulates at their overlap in the central spindle, and from there it arrives to the equatorial cortex of the cell as the spindle expands. Basically in a full agreement with the hypothesis of the equatorial activation. So my story, however, starts in about year 2008. Then Ann Miller, who at the time was a postdoc in Bill Beeman's group, was imaging the activity of RhoA in frog cells that were injected with mRNA for ACT2, the GEF, right? So as a result, these cells were overexpressing active GEF. So to her enormous surprise, she found not only the Rho zones at the emerging furrows, what you can see here, but also a very rapidly moving faint pattern on the cortex of the cells where no Rho activity has been described previously. Some years ago, Bill Beeman at the meeting showed me this movie, and he asked me, "What do you think, Andrew? Does that look like a Turing pattern?" I looked at that and said, "No, Bill, but it does look a lot like waves in excitable systems." So nothing happened for a number of years, but then later on Bill wisely decided to revisit this very curious phenomenon, and he attacked it from a different angle by first imaging the activity, or polymerization of F-actin on the same cortex of these cells. What he discovered was the following. This very complicated, rapidly evolving pattern, sort of shimmering and rather difficult to analyze, as you can see here. However, at a higher spatiotemporal resolution, this pattern can be resolved into the waves of actin polymerization that are moving along the cortex. So the tool of choice to study this kind of patterns is the chemograph, right, which is shown here. So on the chemograph, the unbroken, unperturbed runs of the waves appear as slanted straight segments like this, whereas this type of pattern is representative of the collision and mutual annihilation of the waves. The inhibition of myosin 2 with blebistatin didn't alter anything in this system. However, inactivation of the activity of RhoA, either directly using the Clostridium toxin C3, shown here on the left, or indirectly by injecting cells with dominant negative ACT2, resulted in complete erasure of the waves from the cortex, which basically suggested to us that these waves of actin polymerization might be related to the waves of Rho dynamics that Ann Miller was observing sometime before that. However, it turned out that observing waves of Rho in so-called control cells, which means the cells which were not injected with the extra amount of ACT2, turned out to be a lot easier in the echinoderm cells. So this is, you see a starfish oocyte, and in these cells, the wave pattern has a very strongly cell cycle organized dynamics. So the waves appear at the exit from mitosis from the so-called vegetal pole of the oocytes. They propagate towards the animal pole, as we see now, and they culminate in the ejection of the so-called polar body, which is basically an equivalent of cytokinesis in oocytes. Even a small overexpression of the GFEC2 in these oocytes resulted in a dramatic expansion of the time window all the way from the mitotic exit of one cell cycle all the way to the entry into mitosis in the next cell cycle. Also, what it did, it basically made the wave pattern to be considerably more regular and prominent. So me and my postdoc Marcin, looking at this regular pattern, rejoiced and started applying methods of analysis standard in physics. And what you see here basically is the results of autocorrelation analysis of this pattern, which shows that it is indeed quite nicely periodic in time and very weakly periodic in space with the characteristic wavelength of about 18 micron. So which obviously explains... you are seeing actually raw. This is raw, yeah. The actin, yeah, I was switching, yeah. But I will show you very soon both of them, so you will see both of them. So which basically shows that to observe this type of pattern, you need to have very large cells. And as you can see, our cells are indeed very large. So now, yeah. Which is the source? That's a good question.
Yeah. If there's a spatial concentrated source of the instability, or is it just emerging as?
Yeah, they can emerge all over homogeneously, right? So there is no clear source in space, if that's what you think. So basically, once we knew that both F-actin and Rho exhibit wave-like dynamics, the question was how the two patterns are related to each other. So to our surprise, both in starfish, and now you will see both of them together in two colors. So, come on. Both in starfish and in frog, the two types of waves did not coincide, but instead they followed each other with a fixed species-specific time delay, which is actually nicely seen if we again look at the chemographs, right? If we compare the chemographs, we can see that the raw pulse comes first, after it comes the actin pulse, and then there is a period of quiescence, and then the whole pattern repeats again. So basically this demonstrated to us that the waves of raw and actin are related, they are part of the same wave-like pattern, but the two signals do not coincide. So how could that be understood? So the answer to this question can be again pulled out from the analysis of the chemographs. Indeed, at each pixel of the chemograph, we know not only the amplitude of both signals, but we can also numerically compute the rate of change of either of the variables, right? So here, what you see is my postdoc Marcin built the dependence of the change of rho versus the amplitude of the raw signal at the same pixel of the chemograph, right? So each dot on this scatter plot corresponds to one pixel in the chemograph. So this data can be suitably binned, and average and standard deviation per beam can be computed. So this is the result of this procedure, which shows us that actually the rate of raw growth increases with the amplitude of the raw signal, which basically suggests us that raw auto-amplifies itself. In a similar fashion, it can be shown that raw amplifies actin polymerization, whereas on the contrary, actin polymerization always dampens the activity of raw and switches it basically to the negative dynamics. So, taken together, these data suggest that the dynamics of raw and actin in these cells satisfies formal criteria for the activator-inhibitor system, which is known, of course, to exhibit excitable behavior. Well, Bill and George, of course, didn't wait for any such mathematical interventions. They already charged forward to demonstrate experimentally that F-actin suppresses raw activity, and they managed to achieve that by using latranculin. So as was introduced this morning already, latranculin dissolves F-actin. So the latranculin was released from a pipette, which is symbolically shown here in the corner. And you see the result of that. In the area where latranculin was released, the cortex dissolves and the raw basically goes up through the roof. The activity of small GTPase on the membrane increases dramatically. Furthermore, when they used more, smaller concentration of latranculin, more carefully chosen parameter, you can see that what they were able to obtain, they were able to get a wave dynamics with broader and higher amplitude raw waves. The inverse treatment, when we basically used just plaquenil light to make actin cytoskeleton more stable, produced what basically was expected. The wave activity was dampened and eventually died out. So looking at these wave patterns that George and Bill have been measuring experimentally, Martin and I couldn't help ourselves asking a question. So what are we looking here at? What type of spatiotemporal pattern is this? And has it been seen basically in other fields of nonlinear dynamics? And sure enough, of course, one would expect that in excitable system we should see spiral waves, right? However, as you can see, just by directly observing this pattern, this is not very straightforward conclusion. So we clearly needed a different approach. So now if you take all the points from this image, right, and project them on the plane of two normalized variables, you obtain a cloud of points, right? So instead of using the amplitude variables, here Martin introduced a single angular variable, which is known as phase, and it goes between minus pi and pi, right? So in this representation, the dynamics that you see on the left will look like this. So it's hardly much simpler than the dynamics on the left, but there is something which we can make a conclusion by looking at this pattern, right? So we see that there are colors rotating around particular points in the medium, which are highlighted here by the black arrowheads, right? If I now take a neighborhood of one such thing and magnify it, we will see that in this point all the rainbow colors come together. So all phases and none, right? This is the definition of the phase defect, which is the core of the spiral wave, obviously. So basically this told us that although we cannot see in the amplitude picture the spiral waves due to a lot of noise and the fact that the cores are continuously created and destroyed and they're moving very fast, this is basically the same as a very well-known phenomenon which is called spiral wave turbulence or spiral core turbulence, which is very much akin to the what's happening in human heart during the fibrillation. Well, next we asked following question. Would using a small activator inhibitor model help us to understand the system better? So in our model, obviously, the main player is the raw GTPase, which in its inactive form can shuttle up and down between cytoplasm and the membrane. And once on the membrane, it can auto-activate itself by means of the GEF-ACT2. And the active form then promotes actin polymerization. Once actin polymerizes to a sufficiently large amount, it starts to suppress the activity of RAW, for example, by increasing the rate of inactivation of RAW. Well, at least one possible explanation. In the absence of RAW activity, polymerization of actin slows down and eventually depolymerization and recovery back to the cytoplasm takes place. So we parameterized this heuristic model so that its spatiotemporal characteristics fit the dynamics observed in the ACT2 overexpressing starfish oocytes. So one thing that we saw in these cells, the starfish oocytes, is that there is a characteristic change in the wave pattern which shows this kind of explosion of raw activity around the time when the polar body is emitted. So at the time of cytokinesis, basically, there is this dramatic change in the wave pattern. So we hypothesized that this could be explained by a temporary increase in the activity of ACT2. So this is the behavior of the model with under this assumption, and you can see that the pattern undergoes this kind of transition. If we compare now the chemographs of the two, the model and the experiment, we see that the model captures this sort of characteristic signature of this phenomenon. Though this is not, of course, the proof, but they also have more subtler arguments which basically support this idea.
Can I ask one question?
Sure.
So
Now we have time, so it looks like.
I mean, this is beautiful, okay? Excitable waves, somehow you make the assumption of isotropic processes. I mean, you have
Yes.
isotropic diffusion, but in the preceding talk, which I heard that the actin system is anisotropic, okay? Would this not also be another important element?
Well, this is not a lamellipodium, right? This is not a lamellipodium, so we don't have here organization of filaments according to some kind of leading edge, right? When we visualize actin cortex in the cells, it's finally isotropic. So it's very thick, it's very isotropic. So we didn't worry about any anisotropy, quite honestly. In the first approximation, that was
Coming
sufficient.
back to this question which you had, why do you not see spirals, okay? Maybe the core of a spiral is not possible in an actin system.
Maybe the core is
Yeah,
not
the
possible.
core is a topological, I mean, defect, okay? It's probably not so easy to do with filaments.
Okay, one idea.
Which is somehow related, but it's more about the experimental side.
Yeah.
What, when you say you depict actin, what do we really see? Do we look, how deep into the cell do we look in some way?
We look several hundreds of nanometers. So this is a maximal projection of several planes, right? So-called
this is basically
Z-stack.
all the cortex. It should be
Significant amount of cortex, significant amount of cortex. We've done quite a lot in terms of trying to see what happens deeper. Well, the wave activity, of course, happens close to the membrane. The membrane, of course, is not ideal two-dimensional object. It's a wavy structure and all things. Plus, there is a microstructure in the membrane. But when you project these several Z-planes, you are getting this kind of picture.
And when you say that it's 2D, it's isotropic, then
Well,
I guess that's a 2D
I'm not,
statement, right?
I'm
It's
not
a
sort of
3D.
putting my head on a chopping block, but just by sort of looking at this pattern, we didn't do any very complicated analysis, as you saw. Even
No, but it's just a question what you meant, and if I understood correctly, you mean in the 2D plane.
yeah, yeah, of
Because
course.
I guess in 3D.
Oh no! I never meant anything about third dimension. I understood the question was about 2 dimensions, right? So,
To follow up on this, I mean, in the first approximation, maybe what you see as the actin signal is the thickness of the cortex.
no,
No.
that we know.
Why?
The thickness of the cortex doesn't change. There is a very labile fraction which appears and disappears, right? When this wave passes, this actin depolymerizes. So there is old actin which we see with one method, and there is a new actin which we see much more labile. The new actin appears during this wave propagation and it disappears. It's very difficult to say. I mean, ideally it should be right on the membrane, but we cannot answer precisely biologically where these monomers are. Yes, Hans?
How do you distinguish between old and new actin?
Well, good question indeed, right? When we use these fluorescent methods, what's interesting, and I discussed this with Bill many times, right? And basically, the feeling I get is that luckily, or unluckily, but the fluorescent signal from the reporters that we get is coming pretty much from the new polymerized actin, right? My hypothesis is that the old and sort of static cortex is so covered with tropomyosin and other protective molecules that these reporters, we use two types of reporters. We use LifeAct and we use eutrophin, right? Eutrophin binding domain, and both of them show these waves. We even demonstrated that one binds earlier than the other, which is expected. So basically, all sorts of control experiments have been done to ensure that we don't see artifacts. But this question is sort of open, but my feeling is that this old and static cortex, we don't see a lot of it in fluorescence. However, when we use rhodamine, right, so we fix the cells, we use rhodamine, then we see a lot of this thick and very static cortex. Yes,
just to clarify, yes, LifeAct doesn't bind very well to stress fibers, which are covered with
That was my hypothesis, yeah.
Right.
Yeah, so my hypothesis was that there are old polymers which are protected from all these things. Yes, please.
I'm wondering in the experiment whether your spiral cores are moving around.
They are moving around like crazy.
And what about the model then?
Oh, in the model they are also moving, but it's devilishly hard to obtain identical dynamics. So
Maybe, yeah.
we tried for a long time. We get spiral turbulence here, as if you track these spiral cores, you will see that they are moving, but somehow in real life they are jumping like hell, right? There is probably a lot of noise, which we introduced also, of course, in the model. I don't talk about it, but there is stochastic term here. Yes?
That's what I was going to ask, because the phenomena is fundamentally stochastic.
Very stochastic. So we have rather strong Poisson noise, which is introduced through the actin subsystem. Because we found that if we change basically rate of actin polymerization, depolymerization, assuming that it's an inhibitor for activity, it introduces very strong sort of randomizing action in this model.
But in the model, can the spirals break up? Can you have?
Sorry?
Can in your model can you have?
Can the spiral break up? Oh yes, they do. Yes, they do. Definitely. Yeah. They definitely
so
do.
Where, I mean, where does it happen?
Annihilates or nothing.
No, I'm not talking
Well,
about.
yeah, as you can see, these spiral cores, they die and they are born from the broken ends. So
Sorry, one more question to the model. It's a reaction diffusion model, right?
Absolutely.
So is the actin, is there also diffusion?
actin, we don't diffuse actin. We assume that actin is a non-diffusing variable.
Okay. So this diffusion is only on the...
Yeah, we have two diffusing variables, which is active and inactive raw, and there is actin which does not diffuse. So there are actually three variables in this system. Right. Let me continue. So finally, in the mitotic cells, we found that the wave pattern is not only modulated in time, but it's also modulated in space. So if you look at this cell, you will see something very interesting. The waves appear suddenly all over the cell cortex, and then they're very rapidly extinguished at the poles and shoved towards the nascent furrow, right? So we basically assumed that this is probably the effect of microtubules that are basically collecting the ACT2 from the cytoplasm towards the nascent furrow. So in the model, this is what we implemented. So what you see here is basically a transition from the uniform homogeneous oscillation or excitable dynamics to the situation where all the GEF is being exported towards the nascent furrow. So the system undergoes this transition, and we found that as we drive this parameter, actually our excitable system undergoes transition to a new stable steady state where both activity of rho and activity of F-actin are quite high. So the prediction of that model basically, in the chemograph, this pattern looks like this. So the prediction here is that if we suddenly dissolve the microtubules in this system, then the furrow must disappear and the excitable dynamics should reappear homogeneously everywhere in the cell like shown here, right? So this is turned out to be indeed the case. So what you're looking here at the experiment where there is a multipolar spindle. So you see two crisscross furrows, right? And immediately at the beginning, nacodazole is added to the system. So what you see is first furrows disappear and then the excitable dynamics reappears until the entry to the next mitosis, which wipes out the excitable dynamics, and yeah, so this basically cycles back and forward. So what's the biological significance? What's the biological significance of cortical excitability? Well, we think that it basically explains the sensitivity of the cell cortex to the changes in the position of the cell spindle, right? So the autocatalytic activation of Rho ensures that a new furrow is formed at the position of the new central spindle, whereas the F-actin-based negative feedback ensures that the old furrow is very quickly wiped out at the position where there is no longer excitation stimulus coming. So with that, I would like to thank, of course, all the people who contributed to this project. So that being my research associate Martin in Edinburgh, and of course my collaborators Bill in Madison, George in Oregon, and Ann Miller, who is now an independent group leader at the University of Michigan at Ann Arbor. Finally, since I have some more time, I would like to show you one more movie. If you thought that you already saw everything, you haven't seen anything. So this is one of experiments. This is one of the experiments where basically the cells were manipulated first to believe that they are continuously in mitosis because they have a non-degradable cyclin B, and then there is a pulse of roscavitin coming from the left bottom, which basically stops CDK activity, right? As a result of it, all the cells are prompted to exit mitosis, even though they wouldn't do it naturally. And as soon as they do that, they erupt in this remarkable raw dynamics. As you can see, the wave pattern changes quite a lot throughout the time. It's very prominent, very strong. It's all over the place. It's not something that you saw from the beginning. But unfortunately, we only got to that point at the very end of the project, at the time of almost writing a paper. So with that, I would like to thank you for your attention and take any questions if you have.
So just a question in terms of your noise. Could you remind me where you put the noise and why you put it there?
Where we put the noise? The noise we put in the F-actin term, which describes depolymerization of actin. So we change the rate of depolymerization noisily. And why? This is kind of empirical observation that this is where we get maximal sensitivity of the model behavior to perturbation. If we put it in activation of Rho, we don't get much of the difference, for example.
Yeah. So if I understood correctly, you showed this for starfish and frog, and you used ACT2 overexpression
in both cases
to see the effect.
Yes.
Okay. So would you see this in a normal cell, so to speak?
I showed you a few wild type cells. So at the beginning, the F-actin dynamics was in wild type frog, so no extra ACT2. We can see a lot of actin dynamics, but very faint raw, right, without overexpressing ACT2. In starfish, it's the other way around. We see this explosion of raw, but hardly any actin, right? So when we overexpress ACT2, we see it nicely in both types of cells. So that's basically But of course, our biological laboratories spend months to prove that this can be seen in the control cells.
But you would argue that in principle that's always there in the background, and you have to choose the right conditions to really see it.
Provided you look at the right phase of the cell cycle. And
So following on from that then, you're arguing this is just to position the spindle. So in a cell that is not dividing very fast, unlike these cells, but a cell that's, like a mouse,
yeah,
whatever.
whatever, yes, you're right. Yeah.
Or it's not dividing unless it's wounded, you wouldn't expect to see this?
Well, yes, there are two sort of parts to it. First of all, size of the cell, right? So this phenomenon is, as I show it, it's very large waves. So we are talking about 10 to 20 microns, which is larger than your normal cell, right? Another thing is that as cells in embryonic development become smaller and smaller, the window becomes narrower and narrower, right? So this is presumably because this period when Act2 doesn't know where to go gets also narrower, right? So it's all sort of a big hand waving. We don't know precisely details, but yes, it's basically a luck, I think, that we had these very large embryonic cells and oocytes, and but they're really useful tool now for us to understand the mechanics behind it. Yeah.
Hopefully everybody can hear me. But have you ever made a plot of the concentration of Rho and actin at the same time and see if they do follow some sort of a pattern in which they are oscillating slightly out of phase, like in a predator-prey model, for instance?
Yeah, and of course we did it. We just used a standard autocorrelation analysis and cross-correlation between the two signals, right? So we get a fixed shift between the two maxima.
So one of the hot topics in ecology, for instance, has been the modeling of predator-prey interactions by discrete models or individual-based models, where you can write down a master equation, and from the master equation so it's intrinsically noise.
Right.
You derive the master equation, you derive the mean field limit, and the mean field limit itself has equilibriums that are flat. But then when you see the simulations, what you see is spiral patterns or oscillations, and these are induced by the discreteness in stochasticity of the system rather than as part of the mean field. It's like a stochastic resonance effect induced by the discreteness of the system. So maybe the reason why you're not seeing these full waves is because they might, I don't know, I mean, I really don't know, right? I'm just on very shaky grounds here. It's because these oscillations are maybe a stochastic effect, so you cannot actually form full waves, but you can see these oscillations because of this discrete intrinsic nature of the system, which would be, I don't know if it would be of any value or
interesting idea, right? I would like to discuss maybe later with you how to sort of explore it any further.
So very nice. I just wanted to make a comment. So to me, it has always been very surprising that since you have all those reaction mechanisms that are essentially excitable, that you don't see them in cells all the time. And like in dictostelium, the system I've worked with a lot, you see lots of actin waves and also PIP3 waves and so spirals, and you can get all those dynamic patterns because all the reactions are intrinsically nonlinear. And so also in dictostelium, so if you make very big cells, basically fusing them, and so these are experiments done by Gunter Gieris and some of the other people, you get kind of very nice spirals and lots of things you look at. And I think these waves are basically a general property and they're always there. And in dictostelium, the waves are very important in the propagation and alignment of the motility response in the coordinating systems. And
Definitely.
So I think this will turn out to be a very general property of living systems.
Yeah, we also think that this is quite generic property, right? And the elements may differ, so we don't see any pattern in PIP2, for example. We looked at it, there is no PIP2 waves associated with these waves. Also, our reviewers asked us about myosin. We tested that, also doesn't seem to contribute. But we clearly see the waves of GTPase, which I don't know if you guys have ever looked in dictyostelium, maybe you should.
I have a question. How do you validate your mathematical model given the experimental data?
Validate. Well, that is a very good question that the reviewers usually like to ask when they kill your paper, right? Because in biology, you can validate nothing, right? You can do thousands of experiments, and at best, what you get is data consistent with hypothesis.
But can you
You can never prove anything.
Yeah, but can you reproduce some of the wavelengths? Do you get
No. The model at the moment is entirely heuristic, which means we don't have any kind of constant values, so we can't reproduce them. What we are doing, we are fitting the model to make wavelengths as in the nature. Right.
Short question, what's the typical wave speed you see in the experiments?
Oh, yeah, no, I mean, as you remember, 80 seconds, 18 microns, so you make the division, 20 by 20
We talk about
micron per minute.
per second, or what?
No.
No.
Wait, what did I say? Seconds? Was it seconds? Just a second. Can it be too fast? Can it be too fast? But yeah. So 80 seconds, and we have 20 microns, so about 15 micron per minute, something like that.
No, what I was aiming at is could lateral flow be part of the explanation? I mean, you say you think
Lateral
isotropic,
flow.
so you don't think so, but
Lateral flow. No, I mean, lateral
inclined
flow
filaments
doesn't
by the
quite apply here, because what you mean is what you described, right, in lamellipodium, right? So you're looking at one-dimensional projection in your case, right? In this case, it's a two-dimensional surface.
No, I know, but if you would have a group of filaments, if it would not be isotropic, as you say, if all filaments in a group would look in the same direction, right? Then
we don't know which direction they look, but we suspect that the formins are involved. So it's possible that they grow inside rather than parallel. So we don't think it's up to 3 there, right? But could be formins.
I guess we'll continue over coffee.
It's also